19th century scientists were very interested in bacteria, and there was a great debate about where these mysterious microbes came from. Everyone knew that milk would sour if left in a warm room. Even after the milk had been boiled sufficiently to kill all bacteria, it soon spoiled due to renewed microbial activity. The prevalent theory in the mid-1800s was that bacteria originated spontaneously. The combination of air and food supposedly brought new microbial life into existence. The process was called spontaneous generation, meaning the creation of life from non-living materials. To a young chemist named Louis Pasteur, this notion was absurd. Larger forms of life reproduced biologically. Why should microbes behave differently? Pasteur had observed particles floating in the air, dust specks which became visible wherever a sunbeam entered a darkened room. Was it unreasonable to expect that bacteria might also be transported in this way? The problem was clear, and Pasteur began what was to become one of the classic experiments of biology. Broth of a kind that spoiled very quickly was boiled to kill any bacteria which might be present. The neck of each flask was drawn out and bent into an S-shaped curve. Air could reach the broth, but airborne bacteria were trapped by the curved neck. Within Pasteur's swan neck flasks, the broth remained fresh for weeks, and as it turned out, for years. However, if one of the necks was broken off, the broth in that flask turned cloudy within a few days. Airborne bacteria had reached the broth and multiplied. Pasteur's work showed convincingly that it was airborne bacteria that caused the spoilage of food. His elegantly simple experiment laid to rest once and for all the erroneous concept of spontaneous generation. Pasteur's experiments seem all the more remarkable considering the size of the organisms he was working with. Paramecium is one of the smallest organisms that can be seen with the naked eye. Even with a strong magnifying glass, paramecium is the only organism visible within the eye of the needle. By doubling the magnification, other organisms become visible. The bacteria which paramecium utilizes as food. The bacteria being knocked about by paramecium's cilia average 2 micrometers in length, or about 1/100th the length of a paramecium. Magnified 400 times, bacteria can be seen undergoing digestion within paramecium. Most, but by no means all, bacteria are in this very small size range. Here, a paramecium forages about within a tangle of strands produced by a filamentous bacterium. Large or small, bacteria show very little internal structuring. If organized structures are present within these tiny cells, they are smaller than the shortest wavelengths of light, and therefore cannot be seen by means of the light microscope. Here the electron microscope takes over, but first the bacterial subjects must be prepared in a very exacting way. Initially, the bacteria are killed in a solution of fixative. A centrifuge concentrates the dead bacteria into a lump of cells which is dehydrated in alcohol and then embedded in epoxy resin. The sample is placed into a mold and more resin is added to create a plastic plug. When hard, the plug is placed into the ultramicrotome, a device which uses a glass knife to trim incredibly thin sections from the plastic plug. The sections which float off on a water film are but a few molecules in thickness. Imagine an organism hundreds of times smaller than any which can be seen with the naked eye is being sliced up like a loaf of bread. The ribbons of plastic containing the bacterial sections are picked up on a grid for examination in the electron microscope. The grid is scanned in order to locate the thin sections of bacteria. And there they are. These bacteria are being visualized by a tightly focused beam of electrons. Passing through the sections, the electrons create a shadow picture, which is enlarged by a series of magnets. The final result is recorded on a black and white photographic plate. Even when magnified 50,000 times, the bacterial cells appear to have a very simple internal structure. There is a cell wall, and within it, a membrane surrounding the cytoplasm. However, the bacterium contains none of the complex organelles found in more advanced cells. Another major difference is that in bacteria, the DNA is not contained within a nucleus. Here we can see the strands of DNA distributed throughout the cell. Cellular organisms which lack a nucleus are called prokaryotes. In addition to bacteria, blue-green algae exhibit this primitive condition. All other cells, including our own, have a well-defined nucleus and are called eukaryotic cells. Fossil evidence suggests that the prokaryotes occupied the Earth for the first 2 billion years of its biological history. Only within the last billion years did eukaryotes appear. In bacteria, we are evidently looking at life in one of its simplest and most primitive forms. The rapid motions of certain bacteria puzzled early researchers. Their movements indicated that bacteria possessed a locomotor apparatus, but none could be seen. There was indirect evidence that these motile bacteria were equipped with flagella. For example, when India ink was allowed to seep underneath the cover glass of a bacterial preparation, it became apparent that something was agitating the ink particles near the ends of the bacterial cells. A single bacterial flagellum is too small to reflect visible light waves. However, in certain bacteria, a number of flagella twist together, forming a structure of sufficient thickness to be seen with the dark field microscope. Other bacteria produce motion by rotation. These are spirochetes, relatives of the corkscrew-shaped organisms which cause syphilis. While filming bacteria in a sample of pond water, we found convincing evidence that these primitive organisms can respond to their environment. The subject was a dead paramecium surrounded by a population of motile bacteria, which were apparently digesting the carcass. Watch individual bacteria as they move away from their food source. By means of behavior, these bacteria are able to find and remain in an environment favorable to their feeding activities. Filming these primitive prokaryotes, we had the feeling of looking back through time at one of nature's early experiments in the evolution of behavior. Given an ample food supply, bacteria can multiply with astonishing speed. A few chunks of dry pet food added to a jar of pond water will produce a rich culture of aquatic bacteria overnight. Bacterial reproduction is accomplished by fission. The DNA of each cell separates into two identical genetic packages, followed by division into two new cells. The bacterial cells feed by releasing digestive enzymes which chemically break down the pet food. The nutrients released are then absorbed and used for further growth and reproduction. Theoretically, the rapid growth phase will continue until the food supply is used up. However, long before this happens, the bacterial population will be brought under control by bacteria feeders such as paramecium. Most of our understanding of bacteria comes from experiments which involve culturing these microbes under controlled conditions. Soil bacteria, for example, can be cultured on an agar substrate to which nutrients have been added. Kept at room temperature for a few days, colonies of bacteria have formed on the surface of the agar. The different colors and shapes of these colonies indicate different species of bacteria. These can be separated for identification and study by a technique called cross-streaking. Inoculate a sterile agar plate by making a streak along one side. Flame the needle. Cool and cross-streak. Using the cross-streaking technique, several different species are isolated. Inoculating a new plate from one of the isolated colonies will produce a pure culture of that particular bacterium. There are many variations of this procedure. For example, it is sometimes desirable to isolate and grow several species together on the same agar plate. Soil microbes show an important feature of certain bacterial life cycles. They produce spores. In order to examine bacteria for spore production, the organisms are transferred to a drop of distilled water and fixed by carefully passing the slide through a flame. The preparation is stained with crystal violet dye. When dry, the bacteria can be examined under the oil immersion lens at 1,000 times magnification. This is a field of new, rapidly dividing cells. No spores have been produced. Older bacteria in the colony have developed spores which resist taking up the stain. Spores are resistant to freezing and drying and may spend years in the soil before conditions again become favorable for growth. Since Pasteur's time, scientists have discovered some of the ways bacteria fit into the patterns of life in the biosphere. Some are disease-causing parasites. and these have given the bacteria a rather bad name. However, others have been found to be indispensable to the orderly operation of ecological systems. The next film in this series will explore the role of these essential microbes in the ecology of the planet Earth.